gosh, it's a real privilege to be here. But it's very important to diagnose Alzheimer's disease specifically because the treatment for it differs from the diseases that look a lot like it. The differential, and we'll see why I'm telling you this in a minute, includes things like depression, which can be very hard to tell from Alzheimer's disease. People who are profoundly depressed do not score very well on memory tests and so forth, and their loved ones know that they're not doing very well in that area. And they come in frequently for us to try to figure out, do they have depression or Alzheimer's disease or something else? And that's something that's been very difficult over the years to diagnose, and I'll tell you in a second why we're making some advances in that. Strokes are another one that can look a lot like Alzheimer's disease. And of course, the prognosis for depression is very different from strokes and is very different from Alzheimer's disease. Let me show you in a cartoon here one set of facts that I'd like you to know about. Neurons or brain cells don't actually connect to each other directly. It's not like sticking an outlet in the wall where the electricity goes directly from one piece of metal to another. There's a little space between them. And it turns out to be a brilliant concept for a number of reasons. But the point is that for one cell to talk to the other, they have to release chemicals or proteins that cross over and tell the other neuron what's going on over here. And one of those proteins that's in the neural membrane on one side that gets cleaved off and makes its way across to the other side is called the amyloid precursor protein, or APP for short. It sits in the membrane there, and it has part of its protein is on the inside of the cell, goes through the membrane, and on the outside. It's about 750 amino acids in length. Amino acids are the basic building blocks of proteins. When you eat a piece of meat, you're eating lots of amino acids, and your body takes those in and makes them into all the proteins that we use to live and survive. Now, normally, there's an enzyme called an alpha secretase that cleaves the protein right here whenever the first cell wants to tell the second cell what's going on there. When it does that, then this piece here gets released into the extracellular loop, goes across, and says, hey guys, over on this side, this is what's going on. Now, under certain circumstances, some of which I'll tell you in a minute, this enzyme is not very active at this site. And two other enzymes that have a completely different set of things that they do in cells then will accidentally cleave this protein. And they cleave it proximal to where the alpha secretase does. The alpha is supposed to be there. The beta cleaves it here. Actually, the beta is here, and the gamma is here. And when those two enzymes act, this little 42 amino acid chunk, about 1/20th of the entire protein, gets released. And that thing, it turns out, is very, very sticky. Now, we're all making a little of it, and we have mechanisms in place to traffic it out of our brain. But when that doesn't work properly, it starts sticking to itself. And before you know it, you've got clumps of it with thousands of molecules connected together. And then a lot of other molecules come in and attach to that. That forms the center of an amyloid plaque. When we look in the brains of people with Alzheimer's disease, they're replete with amyloid plaques like this, all over the place. And when you look around them, a lot of the brain cells aren't present there. There's something about these plaques that influence the brain cells around there and eventually results in the loss of them. And that, we think, is what's going on at the cellular level in Alzheimer's disease. Now, an important fact to know in order to understand what's going on in the disorder is that we think that beta amyloid that I just showed in the previous slide is being deposited for 10, 20, 30 years before a person develops symptoms of Alzheimer's. And once they develop symptoms, a lot of people don't recognize them immediately and come in a couple years after that. It might be with some of the items that Dr. Koenig mentioned, behavioral changes, depression, memory loss, etc. So by the time we see someone diagnose them, the disease is probably going on 20 or 30 years. And we have a lot of data to suggest that. That presents a huge opportunity, of course. If we could figure out where was depositing this and why, we would have a 30-year period, perhaps, to try to prevent the development of the disease. As you know, we still haven't gotten very good about treating it once you get it. We haven't figured out yet how to put brain cells back in there and make them work. The idea we're focusing on then is trying to do what we can to prevent the A beta from being deposited and detecting that so that we can study ways of preventing it from being deposited. One of the big things we've been doing then is looking for risk factors that result in more A beta being deposited. And we have about 15 of them now. I'll tell you them briefly. One is traumatic brain injury. If a person gets knocked out one time during their life for 5 or 10 minutes, the risk of Alzheimer's disease goes up about double. The risk of frontotemporal disease, which is the second most common neurodegenerative dementia, goes up about fourfold. We had a paper just a couple of weeks ago published showing that. Another study that Dr. Koenig and I worked on pertains to post-traumatic stress disorder. Very common in the military, but we see it every day in the hospital too. People who come in with heart attacks, get PTSD from fearing that their life is on the line, being in intensive care unit for a week. People in car accidents get it. They actually get it at about the same rate as people in the military, about 22% approximately. We found that that's associated with a 2.2-fold increase in the risk of developing dementia down the road. There's also a whole risk of risk factor, a whole group of risk factors that we previously knew were involved in heart disease and stroke, but it turns out they also greatly influence the risk of getting Alzheimer's disease. That includes hypertension, which causes a 50% increase, diabetes for 1.76, so that would be 176%, hyperlipidemia, elevated triglycerides. Smoking, we know, is a big risk factor for vascular dementia, but it turns out it's also a big risk factor for Alzheimer's disease. It's maybe 3.5-fold increase for vascular dementia, but it's 2.6-fold increase for Alzheimer's disease. Obesity in midlife, meaning if we measure people's waist size in their 40s, there's a predictive risk for Alzheimer's disease 20 years later of about 3.8-fold increase depending on the size. Living in the South, this is another study that Dr. Koenig and I worked on. 18% increase. I think the food is good here, and then it has a certain downside to it. And there may be other factors as well. Familial history about doubles the risk. A lot of people come in and say, well, I've got my mom had Alzheimer's disease, I'm very worried about it. And then, it's reasonable to be worried, but I'll tell you, every other factor that we looked at up here doubles the risk also, or more than doubles it. So family history is not the biggest risk factor. Age is a big risk factor. There's a parabolic relationship between the risk of Alzheimer's disease and age. It doubles about every 5 years from the age of 60 onward. These are 3 that we haven't figured out how to modify yet. The last one is one that we're working on right now. One of our lab partners, Dr. Soto, showed in mice that he can transmit Alzheimer's disease from one mouse to another with a blood transfusion, or if he takes brain tissue from one mouse and gives it to the other. I don't know if that's going to be true in humans. I sort of hope it's not, but since in 95% of the patients with Alzheimer's, we don't know what caused it, we have to look at every possibility to try to understand why it is that people are at risk for it. We know of 3 factors that reduce the risk for Alzheimer's disease. Physical exercise, surprisingly, is one of the most robust reduction factors. Whether you have the disease or before you have the disease, physical exercise is associated with the reduction. There's a variety of reasons for that. When you think about blood flow, free radicals, there's also evidence now for increased trafficking of that A beta out of the nervous system in people who exercise more. I don't know why that is. I rode my bike here tonight just to make sure I protect myself. Mental exercise is also valuable for people with Alzheimer's or not for improving cognitive function. And diet is another one that's been shown to be related. We don't know specific foods, but obviously diet is a very important part of hypertension, diabetes, hyperlipidemia, and elevated triglycerides. So it may be through reducing those that a better diet works. We're not sure about that. The point, though, is that by looking at risk factors and controlling them, we've been able to show in some circumstances that we can reduce the risk of dementia. Dr. Koenig and I, for example, were on a study that came out recently showing that treating blood pressure in diabetics over a 2-year period resulted in a significant reduction in the development of dementia over just a 2-year period. That's really quite surprising, I think. The sort of most interesting thing that I think has happened in my lifetime with this, to be honest, is the development of agents for looking at amyloid in the brain of people who are living still. For those of us who take care of dementia patients every day, we learn to look at CAT scans and MRIs, and we study them, and we study the person, and we ask a lot of questions, we examine them, we look at the imaging, and we scratch our head sometimes, and we just can't tell for sure what the patient's got, and therefore we can't really stratify them necessarily into the best treatment. This whole process of developing an agent has been going on for quite a few years now. There's a lot of technical issues, and finally, about 3 weeks ago, we got a product available that we can use on our patients, and it's just been really exciting in just the first few weeks, and I'm going to tell you a few words about that. This is a normal scan here, and let me show you what we're looking at. In the bottom left here is a CAT scan looking in this direction in a patient, sort of like taking the lid off and looking in the brain there. So this is normal brain tissue, and this is the skull around it. And the brain usually comes right out to the skull there, and that's a normal looking brain. This agent is given in a vein. It's just a couple of cc's, looks like water. You let the person sit in the waiting room we have there for 30 to 45 minutes. Bring it in the PET scanner. It looks like a CAT scanner, exactly the same thing. It doesn't produce claustrophobia like the MRIs. It's nice and open. Person is in there for 10 minutes. They come out, and within a minute, these images start showing up on the computer there. What the agent is on one side, it's sticky, and it goes through your body looking for any amyloid, and it sticks to it. And on the other side of it, it has a radioactive chlorine that sends off a photon that the PET scanner can detect. And it uses mathematical formula to figure out where the photon's coming from, and so it can show you a picture of where it's coming from in the brain. Now, it turns out that the agent binds sort of non-specifically in what we call the white matter of the brain. Those are the areas with the cables that go between the brain cells. The brain cells are all on the outside there. And so what I'm going to show you in the scans here is, we're going to look at the brain cells on the outside and the white matter here. And if there's a differential between them like this, then we know that someone has a normal or not normal scan. This is a normal one because there's nothing in the brain cells and there's a lot of stuff in the white matter beneath it. This is a positive one. You can see from the back of the room that it's very different. There's photons coming from all the way out to the cortex there where the brain cells are. This person's depositing a lot of amyloid and and obviously asymptomatic, then we could say for an Alzheimer's disease. I got the message that I just have a few minutes, so I'm going to show you a few patients very quickly here. This is someone that I agonized over quite a bit. It's a guy who's had depression throughout his life, on and off. He came in with depression again. His wife said, I think he's a little worse than that. I think he has cognitive issues on top of that. We did a scan on him to differentiate between depression and Alzheimer's disease. Not very easy, as it turns out. And a guy like this, it could take several years of treating his depression before you know that you really were testing his cognitive function directly. You can see from the back of the room that there's no dark stuff here and light stuff on the outside. It's dark everywhere. And on the scans here, it doesn't always show up on here so well, but there's clumps of the amyloid in the brain there. And fortunately or fortunately, because I could tell the guy we're going to treat him now, unfortunately, the gentleman had Alzheimer's disease. I was able to tell him this, though, several years before I normally would have, because of the fact that we could do the scanning. This is an 80-year-old person who presented with just mild memory issues and was very interested in knowing exactly what was causing it. And so we did a scan on him last week, and you can see also from the back of the room that there's a lot of amyloid being deposited there. It's really fascinating for those of us that study this, that he has as much amyloid as the previous guy, but is 20 years older and has far fewer symptoms. That tells us there's not a one-to-one correlation between the amount of amyloid and the symptoms that a person has or the location. It's also fascinating that it's often very asymmetric. In this gentleman, for example, there's a lot more in his right temporal lobe than his left temporal lobe. And I found that in all of the first few scans that I've done, that there's a lot of asymmetry there. It's going to be fascinating and important to figure out why that is. If we could figure out why one part of the brain of amyloid is reacting to it so much differently than another, I think it would be really helpful. And the third patient is a very common scenario. This is a woman who came to me from the stroke service. She had a stroke, her memories changed. She hasn't changed in the years since she had the stroke. Does she have Alzheimer's disease or vascular dementia? Well, statistically, she probably has vascular. I followed her for a while. She didn't change at all, but she really wanted to know, am I ever going to change, or now that you guys are treating me for stroke, am I going to be kept from that? I don't know if the lighting is good enough, but there's a white spot over here that's the stroke that she had the year prior. We did the amyloid scan on her last week, and son of a gun, if you could knock me over with a feather, she's got a lot of beta amyloid being deposited. She's in her mid to late 80s, and all she has is mild memory impairment. She hasn't progressed in a year. This is telling us that there's a lot going on there, but again, she's not reacting to the